Hey, welcome back to the Gene to Protein lecture series. This is lecture 3 on RNA processing. First, if you ever hear the term RNA processing, we are talking about eukaryotic cells and not prokaryotes because prokaryotes do not do RNA processing. Why, may you ask? Well, let's remember prokaryotic cells do not contain introns, i.e., non coding DNA. Prokaryotic cells do not have a nucleus. So the process of transcription and translation can occur simultaneously. So there's basically no time to do RNA processing because as soon as that little messenger RNA starts poking out of that RNA polymerase, a ribosome is going to jump onto it and start translating it. So there's simply no time for RNA processing to occur. But because of the separation of both time and place of transcription and translation in eukaryotic cells, then that offers an opportunity for the cell to modify that RNA transcript, and modify it does. Okay, so I love this little diagram up here because, well, what are we seeing? We are seeing the DNA molecule. Okay, so a quick question up here for you guys is, which strand of the DNA molecule is the RNA polymerase going to read? The top one or the bottom one? Think about that. Answer the question. Okay, so once that RNA mature messenger RNA, or once that pre-messenger RNA molecule has been made, then what we're seeing here is that we see all of the exons, and we see they are still, because when the RNA polymerase copies the DNA, it's going to copy both the exons and the introns. And so the RNA now, in order to have a mature messenger RNA molecule, we need to get rid of the introns, the little thin black lines, and we need to put together the exons however way we can. And if you notice in this particular diagram, the very bottom one, out of that one gene, we got 3 different messenger RNA molecules, which are going to lead to 3 different shapes of proteins, which will all have 3 different functions. Now, because they come from the same gene, we say these proteins are related and are members of the same gene family oftentimes, because they're very similar to each other, because they've been made from that same particular gene. But that's the one thing that the eukaryotic cells can do, is they can slice and dice those exons up in a particular gene in multiple different ways. And remember, that is only the first level of protein structure, making that amino acid code. And then, of course, how it folds, and then we can also modify that protein after it has been made. It can be added to another protein, which basically means there are an unlimited number of proteins that can be made from those 20 to 22,000 genes that we do have that do code for protein products. Okay. All right, so a couple more details. And again, all of these steps that we see in eukaryotic genes allows for a more fine-tuned control of the final product and the effects that product has on a cell. And as we'll get into next week, prokaryotes don't quite have that. They can either turn the gene on or they can turn it off, and that's pretty much about it. Whereas eukaryotes, because there's all these steps in there, a much more finely tuned product is going to be made. Okay, so what we see here is we have our transcription unit. This is a single gene. And again, I just want to repeat once again for all y'all, uh, how does replication and transcription translation differ? Replication only occurs, DNA replication only occurs when the cell is going to divide, number one. Okay? Transcription and translation occur all of the time in every cell, except maybe right at that point when we're in the M phase of the cell cycle and all the cell processes shut down. But replication is only for cell division. Transcription and translation is for cell function. Okay. Um, Another thing is that, once again, in replication, we're copying all of the DNA. In the process of expressing a gene, we're only copying one of the 20,000 or so genes that a cell has. And remember, those 20 or so thousand genes that a cell has only represents about 2% of your genome. So it's a very, 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 very small amount that is actually being transcribed and translated. Now, I do want you to take a look at this picture up here and notice that there are four gray boxes labeled A, B, C, and D. Okay. I want you to answer the question and put in, look where RNA polymerase is, what end of the DNA molecule those are. Is it 5' or 3'? Is this 5' or 3'? And on our pre-messenger RNA, is this 5' or 3'? And is this 5' or 3'? Okay, just answer that quick question. Okay, now that you've answered that question, hopefully you got it right. Okay, we see that this is our gene. RNA polymerase, of course, is going to read that gene in the 3' to 5' direction. So this would be the template strand, which would go 5' to 3', and below it would be the coding strand, which is again 3' to 5'. So RNA polymerase always reads in the 3' to 5' direction in order to build a pre-messenger RNA molecule in the 5' to 3' direction. So there's a promoter region just ahead of the start of the gene, and that is basically oftentimes called a TATA box, TATA, which stands for basically because it's heavy in thymine and adenine repeats. The TATA repeats is called a TATA repeat. RNA polymerase, that's a binding site. There'll be a whole bunch of other transcription factors there that bind to that site, telling RNA polymerase, hey, this is the gene we're going to turn on. And it's usually 20 or 30 bases in front of the start codon. There's my start codon, TAC. That's DNA language. Of course, in RNA language, that TAC would be AUG. Okay. And then we have a combination, the transcription part of introns and exons until we hit that stop codon. Again, this is DNA speak at ACT, and of course, in RNA speak, that would be UGA. And finally, just shortly after, it does stop and it disassociates. And now we have our pre-messenger RNA, which consists of both introns and exons. So now we got to get rid of those. All right. So here's just a description of what those things were on the other page. Again, the promoter, that's where the RNA polymerase attaches to TNA, the TATA box. Again, just rich in thymine adenine repeats. Transcription start, where the RNA polymerase starts transcribing the DNA. That's going to be the start codon. The transcription stop is going to be one of the stop codons. Remember, there are 3 stop codons. And then our product will be the pre-messenger RNA consisting of both introns and exons. Okay, so excuse me. So let's dive into those transcription factors. What are they? They are proteins which bind to the DNA and turn on or off transcriptions. Okay, and there are many, many, many different proteins. And there are general transcription factors which are found in all cells. And then there are unique or specialized transcription factors that might only be found in a nerve cell, or might only be found in a fat cell, or might only be found in a muscle cell, or a skeletal cell, or something of that nature. Each cell type has a specific series of transcription factors, but it also has these general transcription factors that are shared by all cell types. Okay, and these will assemble, these transcription factors. You will get a signal, hey, we got to turn this gene on, and they will assemble at a specific series of nucleotides at the start of that particular gene, i.e., the TATA box. Okay, and when all of those transcription factors are there, only when all of them are there, that's when RNA polymerase will come in and bind and read it. Remember, there's really only one type of RNA polymerase, and there might be some other ones, but any RNA polymerase can read any particular gene. Do I know the exact details of how these transcription factors work? Heck no. Do you need to know how they work? Heck no. If you go into serious college genetics research, then you'll get to know them. But right now, just know that there are both general and specialized transcription factors. General found in all cells, specialized found in specialized, cells. So that primary transcript, again, will consist of all the exons and introns. All right. And then other things that have to be done, we have to remove those introns. And then we have to add a 5' cap, and that's basically going to be a modified guanine nucleotide. Has a bunch of phosphates on it. And then we're going to add what's called a poly A tail. All right. And again, that just means many adenine tail. And that many adenine tail can have multiple, 50 to 300 adenine nucleotides on the end of that. All right. And that's going to help determine how long that particular messenger RNA molecule will last before it is degraded, because you'll have enzymes that are going to start eating away at this tail. And the longer that tail is, the longer that messenger RNA molecule will continue to produce the proteins. The 5' cap helps to prevent degradation from the front end, also plays a role in helping to deliver that particular messenger RNA to a specific site in the cell. All right, again, there's our 5' cap. It is a modified guanine, GTP, protects the RNA from hydrolytic enzymes. Our 3' poly A tail, again, protects the messenger RNA, also determines how long that messenger RNA molecule will be active in the cytoplasm, and again, does help export that from the nucleus. Okay, so we got the pre-messenger RNA. We have to go to messenger RNA. What do we got to do? We got to edit out the introns. We have to splice together the exons. And this is a kind of an interesting fact. In higher eukaryotes, that's me, you, your dog, your blue whales, birds, almost all vertebrates and things like that, and probably many plants. 90% or more of a gene can be intron. Whoa, that's a lot. Not exactly sure why, but just because we don't know yet. And maybe, this is older slide. This slide's probably, I've modified it, but I haven't looked at any more research, but it's probably 10 years old at least. So there might be some better information out there. If one of you guys want to go look it up, awesome. Come tell me about it. Greatly appreciated. All right. So how do we do that? We use SNRPs to the rescue. What the heck is a SNRP? It's a small nuclear ribonucleoprotein. There's a mouthful. All right. It's a small ribonucleoprotein. Small nuclear ribonucleoprotein. Whew. All right. So what does that mean? It's made up of RNA and amino acids all stuck together to make up these things. Okay? And what we have is, how does this work? Well, what is a SNRP? A SNRP is one of these small proteins. We have other proteins come together and they assemble. We got 1, 2, 3, got 4 SNRPs in here, and they come to assemble with other proteins, and together they are known as a spliceosome. Okay? I'm sure none of you guys ever had to splice tapes together. Some of you may have spliced ropes together, but back in the day when we wanted to impress a person of the opposite sex, we would make a cassette tape, which took hours and hours, sometimes days to make, because we actually had to splice songs together from different tapes and stick them. It was a pain in the butt. It took a long time. Now all you got to do is arrange them on your computer, hit save, and boom, send off a mix list. I wish that we had that when we were kids, but we didn't. All right, so that's what splicing does. It basically is going to cut the introns, going to use enzymes to cut that DNA at the introns, and other enzymes to splice together, glue together, if you will, these two ends of the DNA. And there's our excised intron. Again, these are going to be nucleotides that will just get recycled by the cell, again, because this is all happening inside of the nucleus. Okay, and there's our mature messenger RNA with its 5' cap, and it'll have a poly A tail and stuff. And again, you can click on this link on your resource slide folder, and that will bring you to a little cool animation about that process. Okay, here's a word too, ribozymes. What the heck's a ribozyme? That is an RNA molecule that acts like an enzyme. All right, before there's a hypothesis out there called the RNA world first hypothesis, where we do believe and have evidence for that RNA was the first self-replicating molecule. Makes sense because RNA, because it's single-stranded, can bind back and fold into different shapes. And remember, different shapes have different functions. DNA has basically one shape, and it's a great shape for information storage and protection. However, RNA can have all sorts of shapes and therefore all sorts of different functions. So we do believe the very early world was basically dominated by RNA. Okay, no organisms yet, no cells or anything like that, just self replicating molecules. Cool stuff. All right. So again, this idea of alternative splicing, again, alternative messenger RNAs produced from the same gene. We can take out the exons and put together take out the introns and put together the exons in multiple different ways. And so we have different messenger RNAs from the same gene, which lead to different proteins. And one of the way I like to think about this is that each exon, and these can be, hundreds of nucleotides long, even thousands of nucleotides long. When that exon is expressed, i.e., turned into an amino acid sequence, that particular sequence of amino acids are going to have a specific shape. It might be an alpha helix. It could be a beta pleated sheet. It could be a straight run. It could be something that's folded over on itself multiple times. All right. And so we could put these different shapes together in different sequences, and we are going to get different shaped final proteins. Okay. And one way we like to call those, sometimes these different shapes might be called domains. All right. So again, discrete functional and structural regions coded by different exons that leads to these domains, and we can put those domains together in different orders in order to have different proteins. So there's our mature messenger RNA. We've taken out the introns and we've added the cap and the poly A tail. And the mature messenger RNA, again, is going to be shorter than the pre-messenger RNA. Because we've taken out all those introns. And as I said earlier, remember 90% of the gene can consist of introns. And plus, due to alternate splicing, it could even be shorter than, be much shorter than that. All right, so the final messenger RNA might only be 1 or 2% of what the entire length of the original pre-messenger RNA transcript was. So just good thing to keep in mind. And that is it for lecture 3. Hope you enjoyed and hope this helped.